If you remember, I told you there are about 300 genes associated with cancer. Human genome has 20 to 30,000 genes, but about 300. You may ask, sir, what is this? Only 300 genes, then what about the remaining genes? What happens is, the genes will ultimately code for proteins, and these proteins interact. So, there is it's a kind of lot of interactions are happening between them. Although you are talking about only 300 genes and so on, but then their gene products will interact with multiple gene products, proteins. So, the protein interaction is also an important thing. Later on we will discuss that in detail. Mainly there are about 4 types of genes that are involved. Although I said 300, but if you want to classify them, then mainly these 4 genes we have to understand. These 300 genes will fit under these 4 categories. Oncogenes. Oncogenes are growth promoting genes. Actually, they hail from what are called proto-oncogenes, about which I will talk about shortly. Then, as I told you, there is always, if something is growth promoting, something else will be growth inhibiting, and these are the tumor suppressor genes. I will correct some of the typographical errors later on. And there are what are known as pro and anti apoptotic genes. So, apoptosis genes, apoptosis regulators, and then last but not the least, the DNA repair genes. They are basically involved in DNA repair. So, we will look at them carefully, and we will also discuss how oncogenes were discovered. Somewhere in 1911, a scientist called Paton Roos, actually I think it is a French word, we pronounce it as a roux. Anyway, we will say as it is written in English, we will say Paton Roos discovered that cell-free filtrates from the chickens affected by certain cancers called sarcomas could be used to induce tumors in normal chickens. So, he did a very interesting experiment, you can see that here. The chicken had sarcoma in the breast muscle. So, he removed these muscle cells and broke them into small chunks of tissue. He ground them with sand. You know, in those days people these days you have homogenizers, something like our mixie, all these machines are there, but in olden days people used to do this by grinding with mortar and pestle. Put some sand to facilitate the grinding, and this way you can separate you can disintegrate the cells from the tissue. Then he filtered them, collect the filtrate that has passed through a fine pore filter. He injected the filtrate into other normal young chickens, and he observed sarcoma in the injected chicken. So, he thought that, but this experiment will immediately make you think that cancer is spreadable. Suppose we inject the cancer cells into other people, you think they will get cancer. No, this is not the case. But in this case, it is because of the viral component that he had in that particular cancer, he was able to do that. He did not know that, but that is what it is. As we know now, we understand that. But normally cancer is not infectious, as I told you. He observed very interesting thing, that is, he saw the injected chickens getting cancer, sarcoma. He did not know what caused the cancer, but predicted that it could be a parasite such as virus. The active agent was later identified as a retrovirus called In his honor, they have named it as Rue sarcoma virus. This was the first demonstration of a virus causing cancer. You remember I told you about the first chemical carcinogenous, in the chimneys, they got the scrotal cancer. That was the first demonstration of a chemical causing cancer. Likewise, here virus causing cancer, this was the first demonstration. It was a retrovirus. Later on, they identified a single gene called SRC, responsible for these cancers. So, this is the first time an oncogene was identified. Actually, this oncogene was identified in this chicken type of virus, right, SARC, but then human SARC also was later identified. In 1981, Michael Bishop, these were Nobel laureates, Michael Bishop and Harold Warmus found that normal cells contain a gene homologous to the virus one that I told you, SARC gene. So, to distinguish this, the cellular origin as opposed to the viral origin, what happens is, see, the viruses, they invade the animal cells or whatever, and then they pick up certain genes, tinker them and then make them into oncogenes. Many of the oncogenes were discovered like that. So, in order to distinguish, so you also understand by this what I said, you also understand these oncogenes are not alien genes. They have been taken from the animals or from the humans or whatever, and then they have been changed, maybe they have been mutated or certain changes have happened, and then they are converted into oncogenes. So, to distinguish this nomenclature was given. V SARC means viral SARC, C SARC is cellular SARC. So, the C SARC is actually a normal one, that is what we call as proto-oncogene. Proto-oncogenes are genes which are present normally, which have the potential to become oncogenes. So, they are called proto-oncogenes. So, this nomenclature came, the idea that the virus picked up the normal genes and modified them upon reinfection could now transform them into cancer cells. Thus, oncogenes are not alien genes, but are actually modified forms of the normal genes. This is a very important concept for you to understand. So, C SARC was called a proto-oncogene. Proto-oncogene means the genes that have the potential to become activated as oncogenes. So, an oncogene is usually in the dominant form. All of you must have studied a little bit about genetics, dominant and recessive. Most of the oncogenes are dominant with a gain of function mutation. A proto-oncogene may get mutated. Again, that is not the only mechanism. I will talk about the different mechanisms by which a proto-oncogene can be converted to oncogene. Mutation is one of them. So that it gains function, so it is called gain of function mutation, and usually it is dominant. So, these concepts have to be understood before we go into the details. So, the idea came, if viruses could induce tumors by mutation of a normal cellular gene, can this be done by chemicals, chemical carcinogens? It was Robert Weinberg, you know, in the US or in England also, Robert, they will call as Bob Weinberg. So, we call him as Bob Weinberg, and his group in Harvard University, they were the first people to show this kind of chemical transformation that could ultimately cause cancer, and they have identified certain genes also. they have shown that DNA extracted from chemically transformed mouse fibroblasts could transform normal fibroblasts, normal mouse fibroblasts. Experiments were done in mice. So, the identification of that DNA turned out to be the RAS oncogene. Later on, when they sequenced that gene, they have initially they have used some, I think, ethyl nitrosourea and they have injected into the fetus, and then they found certain tumors. When they extracted the DNA from those tumors, they sequenced the gene, they found it is nothing but the RAS oncogene. This is how RAS oncogene was previously associated with sarcoma viruses. In 1982, a human oncogene derived from the bladder tumor cell line called EJ, again in his lab only, was shown to be homologous to Harvey murine sarcoma virus, and the cellular proto-oncogene was designated as HRAS. The oncogene carried a certain point mutation, a single nucleotide substitution, which changed the amino acid glycine into valine. Then proto-oncogene was not having this mutation. The oncogene was having this single mutation, point mutation as we call it. So, the glycine was changed into valine. They also identified another RAS which is called Kristen murine sarcoma virus, KRAS, and this is a new member of the RAS gene family which had NRAS, that has no viral counterpart. So, people knew about NRAS, but that has no viral counterpart, but this KRAS and HRAS had the viral counterpart. Further experiments showed that single oncogene, actually Bob Weinberg's lab did this very interesting experiment. They took what is thought to be a normal cell, some fibroblast kind of thing, they transfected with one oncogene trying to convert that into cancer cell, but they did not succeed. They did several experiments, 2 or even more oncogenes were required to convert normal cell into a cancer cell. So, for example, in their lab they used 2 oncogenes called RAS and MYC. RAS and adenoviral E1A, this is another oncogene. So, these 2 combinations, either RAS with MYC or RAS with E1A. So, they discovered that these oncogenes have some kind of cooperative nature, that is, just putting one oncogene may not be sufficient to transform a normal cell into a cancer cell. 2 or more oncogenes are required. So, this is called a cooperative nature. So, this is what these people have proposed. Now, we will step ahead and try to understand what are these oncogenes. As any other genes, they have to code for proteins. So, naturally the question comes, what kind of proteins? Since we are talking about cancer and growth, many of the oncogene products are naturally growth factors or growth factor receptors or cell cycle regulators or even cell death regulators. As I told you, cell death and cell proliferation, they go together. Interestingly, many of them code for protein kinases. Later on, as we will look at it, even earlier also I have told you some of the receptor tyrosine kinases, they are actually growth factor receptors. So, kinases are very much involved in growth. So, protein kinases and above all transcription factors. So, if you want to, there were more than 30-40 oncogenes are known, maybe more than that. Many oncogenes are known, but they will all fit into one of these categories. They will code for either growth factors or growth factor receptors or cell cycle regulators, cell death regulators, protein kinases and transcription factors. We will look at some of them. Initially, when these oncogenes were discovered, many of them had viral origin. So, these namings, nomenclature and all would be from viruses. For example, this one VSIS, which codes for, which is an oncogene, which codes for PDGF, platelet derived growth factor. This is found in fibrosarcoma. And then there is one oncogene called INT2, which codes for fibroblast growth factor 3, FGF3, found in breast cancer. There is another gene called KFGF, which means fibroblast growth factor again, gastric cancer and Kaposi sarcoma. There is another gene called TRK, which codes for nerve growth factor, is found in neuroblastoma. So, like that, these are some of the oncogenes encoding growth factors. So, I am trying to give some examples for each of the types that I just told you. Growth factor receptors, I already mentioned about this. ERB1, because these names are, as I told you, avian erythroblastosis virus, from which this gene was derived. That is why this name is there, ERB1, which codes for epidermal growth factor receptor. That is what we call it as EGF receptor, epidermal growth factor receptor. This is found in the squamous cell, SCC means squamous cell carcinoma of lung and many other tissues. Then there is another one which is called NEU, NE not W, NEU, because neuroblastoma. You remember I told you an experiment was performed in Weinberg's lab, where they injected ethyl nitrosourea and got a brain tumor. From that they isolated this gene. Interestingly, this gene they named it as NEU because it was from neuroblastoma, but later it was found to encode a receptor which belongs to the epidermal growth factor receptor family, second member, which is called because it was found in human tissues, it was called human epidermal growth factor receptor 2, because the first one is EGF receptor. In the same family you find another one, so they called it as HER2, human EGF receptor 2. So, accordingly they have given this nomenclature also, very confusing nomenclature, HER1, HER2, HER3, HER4, there are 4 receptors in that. And interestingly, although it was discovered as a brain cancer and all that, they found it to be overexpressed in breast and ovarian cancers. So, although the name is new, but it is actually encoding something that has to do with breast cancer or ovarian cancer. Then there is another oncogene which codes for hepatocyte growth factor receptor. Hepato means liver. Hepatocyte growth factor means something that stimulates liver cells, hepatocyte growth factor receptor. So, that is called MET. Then there is this FMS which codes for colony stimulating factor receptor. So, this is another CSF, it is called sarcoma. Insulin receptor, by the way, is also a insulin all of you know is a hormone people say, but it can also act as a growth factor. So, that is coded by an oncogene called ROS, not RAS, this is ROS, not RAS. So, this is found in astrocytoma and other cancers. So, this is another example, one table, and then I will give you that. So, we will kind of stop here, because now we have completed this explanation on oncogenes. We will look into the various characteristics later, and then we will also talk about tumor suppressor genes. One by one we will discuss some of the major, because there are so many oncogenes and tumor suppressor genes, we will take some typical examples like P53 for tumor suppressor, RAS for oncogene, something like that. We will discuss that in detail, so that we will be able to understand how they function and so on. So, we looked at some of the examples of these oncogenes. Anyway, as I told you, I will once again rearrange this table, so that you can take one of the tables for your studies. Although I have given several tables in this, I think we can go with this one. For example, it gives you some examples for oncogenes which are growth factors. I already told you about this. Receptors, for example, EGF receptor, nerve growth factor receptor, ERB2 receptor, thrombopoietin receptor. These are all the oncogenes that were discovered. The genes were named essentially from their viral origin. I already told you many of the names. For example, ERBB means erythroblastosis virus. That was the origin. Many of these oncogenes were discovered in viruses, and their counterparts are always found in the animals or humans or whatever. So, that is why that nomenclature came, like I told you, V SARC, C SARC. Most of the cellular genes were actually proto-oncogenes. That is why C SARC is a proto-oncogene, which a proto-oncogene means the gene that has the potential to be converted to oncogene. So, these fundamental points have to be kept in mind. Some of them are GTP binding proteins, some of them are non-receptor protein kinases. I gave you these examples, SARC, BCR-ABL. Some are transcription factors like MYC, Jun, FOS, and so on. Cell cycle or apoptosis regulators also, we mentioned about it, BCL2, cyclin, CDK, and some other examples are also given here. You can go through that. So, these are the different oncogenes. We have also learnt that viral induced alterations may introduce their oncogenes into the cells. That is what it is all about. But then all cancers are not caused by viruses, only 15%, I already told you about that. So, what are the mechanisms by which the proto-oncogene is activated to become oncogene? That is a very important question. So, the next slide is really important one, because it tells you about the various mechanisms by which a proto-oncogene may get converted or may get activated to become oncogene. We will go one by one by simple thing. Let us say this is a proto-oncogene. So, the DNA can have several alterations. This is why I just told you, you must have some background in cell biology and molecular biology, because I will not be explaining what is a point mutation and all that. So, when there is a point mutation, that can result in an abnormal protein synthesis. That abnormal protein may be hyperactive, and thereby it may promote growth. So, this is one mechanism. The other mechanism is by gene amplification. Here, the protein is not really mutated or anything, it is simply amplified. The quantity increases, excess normal protein, the protein is normal only. You can easily understand this. See, Food can also become poison if you take in excess, they say. Even amrit will be poison, they say, if taken in excess. So, the dose, if something is produced in extra large quantities, then that can get you into trouble. There is another interesting example. When we are talking about oncogenes, normally when you artificially introduce these oncogenes into cells, you expect them, the cells to grow. That is what will happen. But if you overexpress, the cells will die through apoptosis. Many oncogenes are there examples. Okay. So, it all depends on the condition. This is something fundamental in biology, you must make a note of. That is, the same gene may do a different function under different conditions. So, that has to be understood. That is why there is a lot of confusion. That is why there are sometimes the drugs are introduced in the market and later on they find some side effects and all that. That is because of our lack of understanding on the molecular mechanisms by which they act under certain special conditions. So, that has to be kept in mind, because this is a very important concept. Then there can be chromosomal translocation. This can produce either excess of normal protein, translocated chromosomes. You know about translocation of chromosomes. A part of the chromosome may get away and then join in another chromosome. It may not be mutual, I mean it will be sometimes mutual exchange, but it need not be equal exchange. That is, if you give this much part of the chromosome to another chromosome, it does not mean that you are taking only that much part of the chromosome. It is not like that. It can happen at random. So, that can result in either excess of the normal protein, like what happened in gene amplification, or the protein that is produced as a result of this translocation could be an abnormal hyperactive protein. That is another mechanism. The other thing is the viruses, although I told you it is only a small portion, but they are also important. Viruses can introduce certain segments into the cells. So, insertional mutagenesis may happen, and that will result in excess normal protein or sometimes abnormal protein, depends. There can be mutation, of course. And then there are local DNA rearrangements. All of you must be knowing these various DNA rearrangements: insertion, deletion, inversion, or so called transposition and all that. And that can result in the production of abnormal proteins. So, these are the mechanisms by which a normal proto-oncogene can become activated to become an oncogene. So, this is a very important slide, as I just told you. So, we will take some examples. Now that we have learnt about the mechanisms, we will have to know certain examples that really happen in many situations. We will go one by one. Deletion or point mutation, this is very famous, may result in hyperactive protein without change in the normal amount of the protein. I already told you. A single point mutation in the RAS proto-oncogene is a very important. RAS is an oncogene, it is a very well known and very important one, can lead to altered gene function. For example, RAS proteins are small 21 kilodalton GTPases. You know what is a GTP and GTPase activity. So, the GTP bound form is active form and GDP diphosphate bound form is inactive state. So, the guanine nucleotide exchange factors increase the amount of GTP, that means they activate GTP bound RAS, and GTPase activating proteins, they actually, once you increase the GTPase activity, you are actually inactivating, because by that action you are converting GTP into GDP and GDP is inactive. So, this is a very important thing. This particular point is very important, because many a time later on we are going to discuss molecularly targeted therapies and all that, where we will be using enzyme inhibitors for therapy. Here you see, if you actually increase the activity, then it inactivates. So here it's a slightly, I will talk about it perhaps later. GTPase activating proteins, for example, if you have more GTPase activity, then it will become inactive, that particular RAS. Okay. So you should if you inhibit, what will happen if you inhibit the GTPase activity? I don't know whether you are getting my point. That is, GTPase activity, if it is there in high, it will actually inactivate, because it will become GDP bound form. So if you inactivate this activity, what will happen? That is, if you inhibit this conversion, what I mean is that if you inhibit this conversion, then it will remain in the active form. So that has to be kind of understood. Later on we will talk about it. You don't have to worry about this. There is a therapeutic application about it. We will talk about it later rather. Let us not worry about it. RAS oncogenes, why we are talking about RAS oncogenes? It's because they are mutated in 30% of the human cancers, colorectal, lung cancers, and several forms of leukemia. In some cancers, such as pancreatic carcinoma, the frequency is as high as 90%, but this is a KRAS form. This is a very well-known aberration in pancreatic cancer. Pancreatic cancer is very painful and treatment also is somewhat difficult. So that is a very important point. Generally in many cancers it is about 30% mutated, but in pancreatic cancer the particular form of RAS, I told you KRAS, HRAS and NRAS, 3 forms are there. So that has to be understood. The mutations found are usually confined to certain specific codons, codon 12, 13 and 61, all result in an increase of the GTP bound form of the protein, that is called constitutive activation. Because we do not know what activated it, in such cases we generally call that as constitutive activation. Constitutive activation means spontaneous activation. We are not able to characterize the exact activators, it happens spontaneously. Such activation, this is an important term to understand, constitutive activation. Later on we will be talking about constitutive activation in the context of many other factors. So we should understand that. Glycine to valine mutation at residue 12 or glycine to aspartate conversion at residue 13, this is very important. Mutation of glutamine 61 to glycine. So these are the changes that happen, that these changes make RAS into an oncogene. So that has to be understood. RAS is an important signaling intermediate in the growth factor signaling, especially involving the receptor tyrosine kinases and the MAP kinase. MAPK means MAP kinase, mitogen activated protein kinase. This is a very important, RAS MAP kinase pathway is very important for cellular proliferation. So that has to be understood. The next example is gene amplification. This you have several examples. What happens is the multiple copies of a gene could be produced. That is what we mean by amplification. So overproduction of the normal protein. Some examples are high level amplification. There is a small error in that, but it's okay, we will correct it. Amplification may cover megabases of DNA and contain variations in copy number across the amplicon. So that has to be understood. There are also what are known as double minute chromosomes. I don't know whether you have heard of that. These are extra chromosomal DNA fragments. So, and homogeneously staining regions within chromosomes contain high level of amplification. That is what I am trying to say is the amplification happens in certain regions of the chromosome. That is what that has been noticed in cancer. The extra copies may be from sometimes from unusual segregation at mitosis. Mitosis after all is not a perfect process, sometimes there can be errors, especially on the extra chromosomal fragments. Then amplicons are frequently found near the fragile sites in the genome. So, there are certain sites which are prone to these alterations in the chromosome. The main example that you must remember, all these are just explaining you how this mechanism happens, but the important one is the example MYC. MYC is an oncogene which is in the, I hope you know this terminology. 8q24 means what it means? Chromosomes, yeah. It refers to the chromosomal regions. So, 8q24 was identified in the acute lymphocytic leukemia. MYC-N, again MYC also has some forms, MYC-N and MYC-L1 and all that, is amplified in neuroblastoma and then small cell lung carcinoma. This is another gene that you may have to remember. This is actually ERB2, as I told you, derived from erythroblastosis virus, and it codes for epidermal growth factor receptor. Later on also I am going to explain this, but this particular receptor, EGF receptor, has 4 members: ERB1, ERB2, ERB3, ERB4. Sometimes they are also called HER1, HER2, HER3 and HER4 because that refers to the human form, human EGF receptor. So, now here we are talking about the second member of the family. As I told you, HERB1, 2, 3, 4, this is the second one. Similarly, HER1, 2, 3, 4, HER1, HER2, HER3, HER4. So, we are talking about HER2. HER2 are sometimes called NEU also, NEU, if you remember in the last class also I mentioned. This is amplified in breast cancer. Usually it is not mutated in breast cancer, but amplified. Amplified in breast cancer. Actually, it is only amplified in 20 to 30% of breast cancer patients, not all breast cancer patients. Only 20 to 30% of them have this amplification, breast and ovarian cancer, amplification of this gene. Then their lifetime is reduced, they will live for 5 years and so on. Of course, thanks to some of the antibodies that have been discovered, Herceptin, now they can be treated, they can live for 25, 30 years or whatever after that. So, that is a very important discovery. There are several translocations. You do not have to mug up all of them, just understand that there are many translocations that have been identified in the various cancers, especially that happens in leukemias and lymphomas, blood cancers. Chromosome rearrangements may result in the formation of a new protein. It can be a fusion protein. After all, 2, 3, I mean like one chromosome goes and attaches with another chromosome. So, that can ultimately encode a different protein. So, new protein, that is called a fusion protein. So, fusion protein could be hyperactive, as I told you. So, this is also seen in lymphomas, sarcomas and leukemias. Translocation of the oncogene, it depends on where it gets translocated also. Oncogene near to the immunoglobulin B lymphocyte, immunoglobulin regulatory elements in certain disorders, T lymphocyte malignancies, may lead to inappropriate expression of the particular oncogene, whatever may be that oncogene. This could result in the overproduction of the normal protein. This is another mechanism. Chromosomal translocations are found both in chronic and acute hematopoietic malignancies and sometimes some solid tumors of mesenchymal origin. One A gene close to the break point on one derivative chromosome is not altered in its structure, but its pattern of expression is altered. The second is that the structure of a gene at the break point is altered. This is a very important one. Commonly, this involves the fusion of exons from two different genes. That is the mechanism to give rise to a novel transcript. See, a novel transcript fusion protein, how it can form? You have the exon, intron, and two exons, if they are fused, then that may result in a particular form of protein. So, that is what is happening. Novel transcript and fusion protein product with altered or novel function. One of the examples is that involves MYC gene in Burkitt's lymphoma, and these are the chromosomal alterations. T8, 8, 14, the alteration occurs between chromosome number 8 and 14. Here it is 24 and 32, like that. There are different chromosomes that can get these alterations, in which MYC gene is activated by translocation to the region of an immunoglobulin gene. In the immunoglobulin also there is, if you remember the immunoglobulin formation, there is this genetic rearrangement. That is the reason why we are able to produce multiple antibodies. So, that mechanism also may give rise to some of the oncogenes. The last paragraph is a very important one. Later on we are going to discuss that. This is one of the best examples, that is the translocation between chromosome 9 and 22. This happens in what is called CML, chronic myelogenous leukemia, blood cancer. This, what happens is, there is a fusion of what is called Abel. Abel is an oncogene. Abel is actually a person, his name is Abelson, and because of his association with the discovery of this, they named the oncogene as Abel, ABL, Abel we say. On chromosome 9q34, with the 50 region, including the promoter of the BCR, that is called a breakpoint cluster region on chromosome 22. So, what happens is, the exchange happens, the details we will study later also. Exchange happens between chromosome 9 and 22 and that results in a fusion protein called BCR-ABL. The fusion protein is called BCR-ABL. It is a tyrosine kinase, and before fusion it will be 210 kilodalton, whereas after fusion it will be something like 190 kilodalton. So, that is what, so the change is, this is often called Philadelphia chromosome because it was discovered in Philadelphia. So, this Philadelphia chromosome is a very important thing. Later on we will talk about, when we talk about molecularly targeted diseases, we will be talking about this again. So, you do not have to worry. All you have to understand is, there is a chromosomal translocation, and that can be one of the mechanisms by which oncogenes could be formed.